Dear students, welcome to. Today we're going to continue the last episode in the chapter 5 and studying the last piece of information in DNA and genetics. And today also we're going to get some refresh and to summarize some most important information on the whole entire episodes, the last episodes and the whole entire chapters. Let's start the lesson. Change in the structure of the chromosomes. In the last episode, we get some information about the human somatic cells and the number and the different types of mutation. And we said that we have two main types of mutation. One of them is called genetic, and the other one is what? Chromosomal. And we said not all the mutation are give us and produced undesirable, but there's some type. Sometimes they are what? Sometimes they are desirable, such as producing kind of plants and some type of animals. And right now we're going to study the change in the structure of the chromosomes. Actually, the sequence of genes on a chromosome changes when what? A part or a piece of DNA is separated from it during cell division. So that may cause what, as we said in the last episode, which is called mutation, and it may be chromosomal or maybe genetics. And it may rotate about 180 degrees around itself and rejoin again the same chromosomes again in an inverted position. But it doesn't occur much more time, but it's, as we said it, they are a rare cases that maybe happen. It's also possible that the non-homologous chromosomes, okay, they are exchange a segment or piece or part of the chromosomes. Okay, segments mean just a small part or small piece of it. That causes what? That the small segment of a chromosome may be lost or gained. So we have here, as we said, the change in the structure of the chromosomes, and sometimes maybe a piece or segment may be separated and inverted about 180 degrees around and rejoin again. Okay, and in other cases, this small segment of a chromosome may be lost or gained. Maybe it's already has been gone, and in some time the chromosome sometime may gains another piece. Okay, it's not necessarily. And if this mutation occurred in reproductive cells, definitely not in any cells, not in the somatic cells, but if this form or this shape of the this sequence of losing or gaining a part or segment of the DNA, if it happened during the reproductive cells, the new trait will appear in the resulting embryo. So we can consider this type of mutation as a what? As a true or real mutation, because it's already transferred to the next generation, as we said. But if the mutation, whatever it was, if the mutation, it occurred and it doesn't transfer to the next generation, we cannot consider this type of mutation is what is a true mutation. This kind of mutation is called gamete mutation. So as we said, it may happen on the side of the genes, and it may be happen to the chromosomes, and if it happened to the what? The gamete itself, we can call it gamete mutation. It also occurs in living organisms that reproduce sexually. Okay, I mean it's it's not necessarily to happen for those living organisms that they are reproduces, that they are reproduced asexually, but they are occurs for the living organisms that reproduce sexually. Mutation also may occur in somatic cells, where the sudden appearance, I mean it's substantially something that appears, of the symptoms occurring in the organ whose cells mutated. So the different types of mutation, it may be happens during the cell division, formation of the embryo, and also it may happens after the life, in the middle of the life, starting of the life, whatever. But it also the sudden appears for what? For the infected or the mutated one. And this sort of mutation, this kind of mutation, is known as what? Somatic mutation. So till now we studied the genetic mutation that occurs to the genes or part of the segment of the gene, chromosomal one, also reproductive one, and also we said the somatic mutations. Somatic, it means that happens to the body cells, okay? Any cell in the body except the reproductive cells. It is more common also in plants that are the propagated vegetatively, where a new branch may grow from the normal plant having a new trait. Okay, so it happens most commonly for the plant, okay, which is propagated or which is divided vegetatively. As we said that we have a different types or kinds of asexual reproduction for the plant, like vegetative, like cut, and so on. We have a different types of it. And we have also the tuber. This branch may be cut off and propagated vegetatively if the trait is desirable. What does mean desirable? Again, we have two types of mutations of we need it or we do need it. Desirable, that we passionate to get it, we need it, okay? We demand to get this type of this kind of fruit. On the other hand, most of the mutation in humans, they are undesirable. As we said, a lot of syndrome like Turner, like the autism itself, a different type, they are undesirable. But in case of fruits, sometimes they are what? They are desirable. I mean, we need it. But what about the origin of the mutations? I mean, from where it come from? We need to study the mutation details and to get much more information to understand why there are some chromosomes or some nucleotides, they change their behavior inside the cell to give us the mutation or undesirable trait. Mutations may be spontaneous or induced. So, as we're going to say the types of mutation, we have four: genetic, chromosomal, and somatic, and the last one is, which is reproductive. But under the title of from where the mutations come from, I mean the origin of the mutation, we're going to study, we're going to divide or classify the mutation into two main types: spontaneous and induced. One of them by main and the other one under effect of circumstances or under effect of the subjected to the environment. The spontaneous mutation, they are mainly originated by itself without any human intervention or without any human interference. And also, it is rare in all organisms. What does it mean rare? It doesn't occur, it doesn't happen, okay, frequently, but they happen occasionally from time to time, okay, rarely to happen for most of the living organisms. This is the spontaneous one, okay? But what about the induced one? From its name, the induced one by the human, okay? I mean the first one by the interference or intervention of the human. The other one, and also the spontaneous one, it is due to the effect of certain environmental factors around the living organism at different environmental circumstances, like what? Like radiations, like subjected to a different rays, like different chemicals, like a lot different factors. Okay, they are called what? Spontaneous mutation by the effect or by the action of the environment or circumstances without any intervention of the human. Such as what? Subjected to the ultraviolet, like cosmic rays. Cosmic rays, they are rays that comes from the outer space, okay? And the human, in most of some cases, the human body, when the human body cannot adapt or cannot resist this change, the spontaneous mutation may appear or may not. And also different chemical compound to which the living organism is exposed or to subjected to it. So it's not necessarily to happen for by a definite type of rays, but we have a lot of reasons. Okay, we have variant reasons for occurring or happening the spontaneous mutation. As a result, as we said, it was from the circumstances. The spontaneous mutations also play an important role in what? In the evolution of the living organism. Okay? Pre-existing structural defenses. For example, and also the induced mutations. Mutations which are induced by man to produce the desired changes in the traits of a specific organisms. So, as we said that the origin of the, as we're going to study the mutation from the side of its origin, we said we have spontaneous and the other type, which is what? The human effect or that responsible by the human, induced one. For desirable or for to get the demand traits. For this purpose, man uses some factor that are found in nature, like what? Like gamma rays, like ultraviolet rays, to affect on some type of plants or some type of living organism to get a desirable. He also may use some chemical like mustard gas, okay, and like choline, like nitrous acid, and a lot of examples of chemicals. And such a treatments cause atrophy and death of growing tip cell in the plants. He used also most of these chemicals or this type of induced mutation to get rid of what? To remove the growing tip in the plant cell. Okay, but new tissues, okay, are regenerated underneath the dead cells, and these new tissues contain some polyploidy cells. As we said, the polyploidy character, they are desirable. We have a lot of them, okay, and they give us a plant with a desirable trait. Sometimes the fruit may be the giant fruits, okay, and sometimes they are delicious and they are more bright in color and condensed in color, as the human wanted that. Most induced mutations produce undesirable traits, but when the man selected from them the useful ones, it's gonna be desirable one. For example, some useful mutations may produce fruit trees with high sweet and seedless fruit. Do you imagine that? Sometimes you need what? To get seedless fruit. I mean fruits without seeds. So this is kind of mutation. But with the human induced mutation, to produce crops or fruits without seeds, it's gonna be a benefit for the human. It was also possible to induce what? Mutations in a microorganisms, I mean the unicellular living organisms, like viruses, bacteria, and so on, to get much more benefit from them in the experiments in different laboratories, and also to make a lot of experiments for the next time in the medicine, such as what? Such as penicillin, okay, which is capable of producing a large quantities of antibiotics. You know that penicillin has a lot of functions inside our body. So by inducing mutations and using these techniques of making and getting the desirable traits, we can produce a much more or large quantities of penicillin or antibiotics for the human. This is a kind of what? Desirable traits, desirable mutations. Right now, we're going to get some refresh and to try to remember some points about the DNA. Okay, and tell me, this picture, it represents what? Yes, the structure of the DNA. As we said, the DNA, it consists of two main parts or two sides. It looks like as a ladder. The sides of the ladder represents what? The nitrogenous bases or the carbon or the ribose sugar. Yes, the ribose sugar. Excellent. And also we said the DNA, it consists of a structure unit. They are called what? Perfect. They are called nucleotides. And each nucleotide, it consists of three main parts. One of them is called ribose sugar, or 5 carbon, or sugar that consists of 5 carbon, which is called ribose. Ribose means here 5. And also we have the phosphate group, and finally we have nitrogenous base. The nitrogenous base, it might be one of the 4 bases. Each nucleotide has just one. It may be adenine, or maybe cytosine, thymine, and guanine. And to remember also that adenine links or combines with thymine by 2 hydrogen bonds, while guanine, it links with the cytosine by 3 hydrogen bonds. And also the DNA, it doesn't remain as it is, but it's going to twist together, as we're going to see in the next picture. We have here, for the DNA replication, we need some enzymes to separate the condensing of DNA, and other, they work to replace or to add the new nucleotide or new pieces. Okay, here that we have the first step for the ladder, or for the double helix. We need to what? We need to separate it. Okay, we need to separate between the two sides of the DNA, of the helix. Okay, by using what? By using the DNA replicase. While the second picture here, we have what? The third one, we have the another enzyme. It starts what? To adding the complementary side for the other side, and each one is complementary to other side to form the complete sequence, or to complete the template of the other side. Okay, and also here, this is what something that to show us, just to remember you, that the DNA in eukaryotic, or in the cell of the eukaryotes, it's represented in form of chromosomes. Okay, and these chromosomes starting appear in what? In the prophase. As we said, the cell division, it starting by what? Prophase, and after that anaphase, metaphase, and finally the last one, which is telophase. The X shape or the crossing form of the chromosomes appear in the prophase, and after that the condensing and forming these crossing shape or X shape of the chromosomes. To repair it, okay, or just to replicate it, we need to unwind the condensing of DNA, and that occurs or takes place via a different number of enzymes. One of them to unwind the crossing or the twisting of this DNA to give us the chromatin, okay, or to give us the nucleosomes. And in each twisting, as we said it before, that we have about 10 pair pairs, okay, in one turn, each one turn. And after that, there is another enzyme to twist also, or to replicate the nucleosomes to show the condensed DNA. And also we have the chromatin, which is the main structure, or which appears, the DNA appears condensed, and by also unwinding or untwisting the DNA, it appears in form of chromatin, and this chromatin actually is combined with the histones, okay, in different points. Finally, it condenses to form the form of chromosomes in prophase to start or to prepare the cell for division. And actually, in two different types of the division, both of them, okay, the mitotic division and meiotic division, both of them, the cell prepares itself by duplicating the amount of genetic material or DNA, and after that the chromosomes appears in form of the genetic material or DNA appears in form of chromosomes starting in the prophase. Let's also to get some revision on the other non-bonding DNA. Okay, as I said before, the settle of DNA in karyotic genome, it contains a great deal or great number of non-bonding DNA, and also going to study the repetitive DNA. And to know its function, till now it's sometime known and sometime unclear. And before DNA sequencing became possible, okay, the DNA amount that carry the large amount of genome. So there is a little relation that came between the organism or the number of proteins which is produced. And remarkably, a little of the DNA of the plants and animals actually codes for the proteins. So we have here some differences between the DNA in prokaryotes and eukaryotic. For example, the largest known genome belongs to one of the amphibians, which is called salamander, and it contains about 30 times the amount of DNA more than which is found in the human cells. So it's not necessarily as a kind of evolution that the DNA is large in amount by the higher vertebrates, or in the higher vertebrate or in the lower vertebrates, or even in any other species like amphibians or birds. Okay. Although they produce fewer proteins, less amount of proteins, and sometimes the DNA acts on keeping the chromosome's structure itself to be stable. And there are some regions of DNA, it acts as references, or it acts as starting point to the places which the messenger RNA synthesis should start, or it should start or begins its work from it as a kind of reference or kind of remark for the mRNA. And these regions are very important in the synthesis of the protein, because if there is no these references or if there is no these remarks, the mRNA, it doesn't work, it doesn't start. Right now, we're going to study the mutation, okay? And we said we have actually 6 types of mutations. The first 4 related to what the hereditary, like we have the chromosomal, the genetic mutation, and also we have the reproductive and somatic. And if we're going to study the origin of the mutation, we're going to study 2 types. Okay, the first type related to the human, and we call it induced. It's already carried out by human for getting desirable traits. And if they are happen under effect of environment or circumstances, we're going to call them what? We're going to call them the spontaneous mutation. Okay, so the certain traits which leads to change in the character or the traits of the living organism, this is the meaning of the mutation. The mutation also considered true, they are real mutation, if they are transferred to different generations, from generation to the next and so on. But if they didn't, so they are not considerable as a mutation. We have also to distinguish or to differentiate between the mutation which is resulted from the change in the structure of the gene or the chromosomes of the hereditary factor, and the change which results from the environmental effects. And those who's happen under the environmental effects, they are called spontaneous, and actually they are very rare mutation. Yet they are sometimes as a result of segregation or recombination of genes together. The accumulation of the factors or accumulation of the characters. But most mutations give us what undesirable traits that we do need it much more, such as what, like the deformations in human, okay, or in human, like most of the syndromes, like Turner syndrome, like autism, and so on. Mutation in the planes also, they are actually, sometimes they lead to sterility and deficiency in crop, but the most cases, the planes can give us kind of desirable or passionate traits to get some benefit from it. But in very rare cases, as we said, the mutation leads to some desirable changes. For example, as we said, we have some information about the American farmer who found that the sheep which has a curved leg is a kind of a desirable trait, because it couldn't jump or it couldn't pass the fence and to damage the crops. And man also tries all possible scientific ways to find it, to induce the desirable traits or desirable genes artificially. For example, here, as we said, we have an American farmer. He discovered that one of his sheep had one short curved legs,